We are in the next chapter, and we're going to talk about predation and parasitism. And now we're moving out of microbes. We'll have some microbes involved because I can't bear not to talk about microbes, but we're also talking about macroorganisms as well. And we'll talk about being a detritivore and an omnivore, how organisms adapt to predation pressure. So we'll go back to that from the last chapter from 19. Now we're in 20. Adaptations of predators, non-lethal predation. So predation is not always lethal. It's really important to consider that because there's a lot of examples of non-lethal predation. Trophic levels in food webs and food chains. And we'll talk about the trophic cascade and other theoretical community ecology concepts at the end. And so we'll start getting into community ecology and how predation is related to that. So many organisms are omnivorous and detritivorous. So this is a stream food web. And it's a web, not a chain, right? So the web being all the interactions in a chain, just being primary producers, primary consumers, secondary consumers, etc. And it's not super well resolved on the bottom. It sort of just has 5 categories, and probably there should be more, detritus, biofilms, and diatoms. It's just the goo living on the bottom, right? And so maybe organisms are more specialized than that, but it's just the invertebrates here. So there would, or for example, be predatory fishes involved and some other things that are not on here. But what we can see is if you just pick somebody out, you pick out this midge larva, it's eating diatoms, it's eating other algae, and you pick out Hematopsyche, this Trichopteran, a caddisfly larvae, right? And it's eating fungi, it's eating detritus, it's eating other algae. So it's eating a bunch of different food sources, and that's what is called omnivorous. And many of these things are just eating detritus, and fewer of them maybe are eating litter directly. And here's an example on the top of one that's, so these at least are things we talked about before, are still sort of feeding basal resources. Sort of basal resources in that, detritus could be anything. It could be any of these organisms that died and fell down on the bottom, right? So it could be really from eating from any trophic level. This Rhicophila fuscula, though, is eating a bunch, is a predator, right? So it's eating all these things. But also, you'll notice that the arrows are going from detritus and biofilms and from leaf litter. And so it's able to eat at multiple levels. It's able to eat some of these things. Let's see, some of these are predators. A few of these are predators. It's able to eat predators, and it's able to eat invertebrates. So it could maybe get predatory invertebrates, and it's certainly able to eat primary consumers. Most of the food primary consumers. But you can see this compartmentalization, that things are specializing on different things. And yeah, so the world's a complex place, let's put it that way. Most stream organisms tend to eat at multiple levels, really. And one of the ways that we know this is from using stable isotopes to document food webs. So this is a bit of a recapitulation of your laboratory, and you've sort of had this already, but it doesn't hurt you to hear it again. The most common isotopic ratios are 15N to 14N and 13C to 12C. And these are stable isotopes. So elements have multiple forms and different numbers of neutrons in them. And nitrogen has a highly radioactive form, which you don't want to mess with. It has a very short half-life, but most nitrogen in the world is 14N. And just a few parts per billion are 15N naturally. But it varies, and so you can use that as a tracer for food sources. Different food sources have different ratios. And C13 and C12, same sort of thing. Again, there's also C14, which is a radioactive isotope that's used for dating purposes and things like that, other scientific purposes. But you can do it with a stable isotope. They use mass spectrometers. A mass spectrometer takes the molecule and shoots it in a magnetic field and deflects it. And the heavier it is, the less it's deflected. And so you can have a sensor, and the lighter ones go further and hit it there. The heavier ones don't go quite as far. I'm sorry, the heavier ones go further. They're not as deflected as far, and the lighter ones are deflected less. And there's other tricks you can use, light and stuff like that, to measure the ratios. And the key point is that you are mostly what you eat, but nitrogen, it fractionates. So essentially, it's harder for you to excrete and get rid of N15 and N14 in your body, and so organisms tend to become more and more enriched in N15 as you move up the food web. And roughly 3 parts per billion, 3 Del units is per trophic level is what we see with nitrogen. And organisms can track the carbon ratio in their food. So they pretty much just match, and there doesn't seem to be the same fractionation. It's just because of the different biochemistry that's involved. One of the really important things about this, well, actually, I'll ask this as a question of the day. Why do you think that it may be misleading to just rely on gut contents to decide what an organism is using to grow on? Okay, so why do you think it might be misleading to use just gut contents, like rip open the gut of the fish and say, oh, there's, you know, this midge head and that fish bone and blah blah, and that's what's allowing it to grow. Yeah, so we're getting some really good answers here. You know, some things might be eaten by accident. So if you're a snail that's grazing stuff off a biofilm, maybe you'll get sand particles, right? So the sand particle will be in your stomach, but it's not something you're going to digest. Maybe, you know, that's an extreme example. Some things might be hard to identify, right? So the things that are digested quickly or disappear quickly, and they may be the primary food sources, right? So you, and then what if organisms eating detritus? You know, there's a big amount of detritus. Some of the stuff might be incidental, like, you know, for example, trout in high cladophora blooms in the streams, their guts will be packed with cladophora, but what they're doing is they're trying to eat the invertebrates out of cladophora, and they just incidentally get it in it. They can't process the cladophora very well. In that case, they start smelling like algae, and people won't eat the fish anymore either. But yeah, so there's a lot of examples like that, and some really good answers there. So it's a nice tool. It's not always clear. It's sometimes kind of hard to parse food webs out. And here's 2 examples, one from a stream and in Lookout Creek in Oregon, which actually just had a forest fire up there, but didn't do huge amounts of damage, and Toolik Lake up on the north slope of Alaska in a lake. And we see that the Del values of C13 here vary between tree leaves, periphyton, and fine particles, and the N ones do too. The tree leaves, because they're photosynthesizing up in the air, the diffusion rate of carbon into the CO2 into the leaf actually fractionates the CO2. The periphyton, also the diffusion across the diffusion boundary layer, fractionates it to a degree. So that explains why the C values vary. The nitrogen sources might be different, the Del nitrogen point. So this is parts per thousand, right? 0 over 2 zeros is not a percent, parts per 100 is parts per thousand. And we see that roughly 3 units higher primary consumers are. They're about 3 higher than tree leaves and about 1 higher than periphyton. So in this case, you kind of say, well, maybe they're eating tree leaves. Okay, but the other thing is, and the predators are definitely 3 higher. So they're probably eating primary consumers, and that's pretty good. If you look at this though, there's something missing, because I said these animals are following their food sources, right, and carbon. So the predators are lining up really well with the primary consumers, but there's some kind of primary consumers, there's some food sources missing here, and it's got to be something that's over here. or there's something that's at least right here that, and they're not eating any of these things, right? So that's because it's supposed to line up on the x-axis here, and it's supposed to be about 3 units here. And there might be something missing down here too in the nitrogen. Let's compare that to, we have a similar problem, but you know, similar situation where we have periphyton in the lake, phytoplankton in the lake, carex, which is a epiphyte on the emergent macrophyte on the margins of this lake. So we're missing something for the copepods that are eating the phytoplankton, but they're about 3 Del units high. But there's something over here, you know, maybe some other terrestrial vegetation or something, but there's not a lot up there. So it's not clear what's there, but it is about 3 higher than these primary food sources. And the fish is about 3 higher than the copepod, so it's probably eating zooplankton. And it's sort of lined up, but again, it's probably missing. The last thing on this graph is that because you have error in your estimates of N15 content and C13 content, there's error bars around each of those. That's those error bars are suggesting. That's what those two-way error bars are suggesting. So you'll see this in the literature a lot, whether you're managing fishes or doing ecosystem food webs or whatever. A lot of times what you can do then is do a comparison of the gut contents to this and sort of sort things out. It does lead to some good insights, and there are additional sulfur and oxygen isotopes, hydrogen, that can be used, and you can resolve things better the more isotopes you used. And so that's another way to approach this. Okay, so this is just a redo of a slide that was back in the microbes. And remember, you can, if you're being preyed upon, you can evolve different ways to get around that. Mechanical, you can be either too small or too large. You know, it's hard for people for anybody to eat the largest fish in the stream, right? That they're going to be the top predator, unless there's a heron or something that comes in from outside. A lot of animals will make spines, or you know, turtles have shells. Those are all mechanical things that you can use. With the fishes, a lot of times it's getting yourself grown up past that larval stage where you're really vulnerable to being eaten by everyone and getting big enough that you're not. Chemical, you can be toxic, and you know, a lot of microbes are toxic, but also animals can be toxic. So for example, anybody know of a amphibian tadpole that's toxic and can be around fishes? It's one that's invasive in a lot of places, actually. It's one of the few tadpoles you'll find when fishes are in the water. It's a big frog. Hint. Bullfrog, good, Aaron, got it. Yeah, bullfrog tadpoles are toxic, so most fishes can't eat them. You can also be poor quality. So we've talked about Cladophora a lot because I love Cladophora. Cladophora is a lot of cellulose and not a lot of, you know, good stuff inside. So you got to really be hungry to eat Cladophora. And so that's an example of a sort of a poor quality food source. And then there's a whole bunch of behavioral. So the eukaryotic, the animals, are really kind of boring as far as chemical stuff. I mean, there's a few chemical defenses and such, but their metabolism is pretty much all the same. You know, there's just boring respiring, and you know, there's no denitrification or nitrification or any of that stuff going on. It's all, you know, really kind of ho-hum on that side of things. They just don't have the genomic diversity that the bacteria and the archaea do, but they have behavior, and the behavior is where they excel. And there's all kinds of different ways they can use escape. They can have chemical, they can have hydrodynamic visual cues. They can do all kinds of things to get away from predators. And you can escape by reproducing. You can escape by more quickly by going dormant. There's all kinds of different ways you can use chemical cues to get around predation. So here are some examples of a couple of invasive species of zooplankton. And part of the reason they're so successful is that they are they have these giant spines, and it's really tough for the things that eat zooplankton to eat them. And the one on the left is Daphnia lumholtzi, and you'll be able to find that one in reservoirs around here. And the spiny water flea hasn't spread as much, but you can just see, I mean, this thing is, it just doesn't even, neither of these look very appetizing, but this one looks like you're just eating a big thorn, basically. So remember, many predators are gape limited. They just can't get their mouths around this kind of thing. And so that gives them an example. I believe these are ones from Africa and ones from Asia. But don't quote me on that. They're definitely not from North America. Here's a behavioral escape from Daphnia. And if you go out and sample, you see that the purple are the high concentrations, and the reds are the low concentrations. So when we look at this, we have at nighttime, we have these really high concentrations of this Daphnia up here in the top 20 meters of the lake. During the day, when it's light out, there's almost no Daphnia, and they're all down in here. Right? So the question is, why would zooplankton move deep into lakes during the daylight? Can't handle the sun. That's possible. That's a good one. We talked about photo inhibition, but they could use carotenoids, right? So we do have animals, zooplankton in the ocean and in ponds and stuff that collect carotenoids from the environment, from the algae they eat, and use them to protect themselves from the sun. So from UV and such, and the free radicals. So that's probably not the answer. Okay, we're seeing, harder to be seen to avoid predation, right? So many fishes are sight feeders, and they can see something as small as a large Daphnia. And particularly like larval fishes, that's a lot of what those things are eating, right? If they're out in the open water. So the Daphnia, why does the Daphnia want to be up in the surface, right? What does Daphnia eat? Phytoplankton, good, right? So the phytoplankton are going to be in the surface because that's where the what is? Light. Good, Cassie. Awesome. Right, so light's there. The algae are growing there. They don't want to be down deep, but they got to stay up there during the night to get to the next day. So the Daphnia is coming up, snarfing them, going down. The other thing that Daphnia can do, and this is, they're able to escape predators also by swarming and by running away from big dark objects. So they just really swim away. This is why we use clear samplers for zooplankton, like the Schindler trap is clear, right? And if we were using a Van Dorn bottle, it was clear too. And people have done the experiments where they take a clear one and a dark one, and they go down into the same and take multiple samples from the same depth, and the clear ones give you higher numbers than the dark ones. So Daphnia has all kinds of ways to get around predation.